Okay, welcome back. This is our last official class meeting. And today we're going to talk about two-dimensional stability, but we're going to really start by having dry runs for people's presentations. And so we're going to have them in order: Ibraheem, William, Pedro, Elmer. And then I would like to talk a little bit about stability theory in two dimensions. And we may wind up having to skip, depending on how long people's presentations are, we may have to skip some of the details, but you'll have the slides and at least you'll have heard some of the concepts. And something that was just asked about was that I've asked you to review each other's presentations. And so what I'd like you to do, we don't have too much time since things are really due a week from today, is in the next 2 days, I would like each of you to meet with one of the people that I've assigned, have them do a more detailed presentation where you do a more detailed review of their project, and then write up a set of suggestions, and then get it to them in time that they can incorporate those for next week. And I realize that that's a sort of a tight schedule, but there's no real way around it because of the way the semester schedule works. I'd like to have had two weeks between the initial presentations and the final presentations, but we're stuck. And next week, for the final presentations, I will check to see if this room is available if you want to do it in person, but if it's not, we'll have to do it online if this room is being used for an exam. So I'll have to find out about that. So it'll be at the regular class time. Any questions about that? William?
I saw you sent us different PDFs to fill out. Do you want us to be doing that during class and after?
So William says I sent out some PDFs, and do I want you to fill them out now or later? So again, this is something we've done traditionally and maybe doesn't work quite as well in a small class. During the presentations, and this is really more for the final presentation, but you can do it also for the initial presentation as well. I gave you a rubric, and I should probably ask people to print it out first, where you can make some notes for general comments about your classmates' presentations. Those aren't expected to be very detailed, just the kinds of things you notice during the class. So there's a typo here, this wasn't clear, this was exciting, little things, the sorts of things that you could write up in 10, 15 minutes that you could hand around to the class. And then there's the rubric for a more in-depth review when you meet one-on-one with people. And that's a more serious review. But it's a bit different. One of them is if you have comments from everyone about what they thought of your presentation and ideas. And the other one is a more in-depth review by one person. And again, in a small class, you don't have as much of a distinction. But if we had 15 people, I would go through and aggregate all of the comments from the short comments for the in-class work, and then you'd have the single longer presentation. So, okay. Again, the goal is to help each other improve your presentations and your project. All right, so I have the order as Ibraheem, William, Pedro, Elmer, and our first up is Ibraheem.
So, like I said, I still haven't put this in presentation mode, and I'll let you guys know what parts of the project I still need to incorporate as I go through in the last final week. But basically, just as a reminder, the project I'm doing is modeling predator-prey interactions in a more complex way than just a simple Lotka-Volterra model, basically by adding and building on it with I guess different, more nuanced. Okay. So basically, we're going to look through the Lotka-Volterra model, and then we're going to slowly be adding more nuanced interactions between predators and prey, so that we can more and more realistically simulate a real world ecosystem. So let's take a look at, I guess, to start, just the simple Lotka-Volterra model. And this is basically described with a function x of t. That's the prey population over time, and then y of t, which is the predator population over time. And the most simple version of this equation has just 4 parameters, a, b, c, and d, and it's described as the change of x is this function and the change of y is this function, with a being just the intrinsic growth rate of the prey population, and then b being the negative relationship that predators have on prey, and then C is the intrinsic decay rate of the predators population, and then D is the positive effect that prey have on predators, and this is best understood as like the predation efficiency. So the more efficient predators are at capturing prey, the greater D will be, and this will ultimately increase predator population. So below, we'll just take a look at just this simple Lotka-Volterra model plotted. And this is basically, as we've seen in class, it's just a simple scenario with an equilibrium constant, the predator and prey oscillate based on their initial starting conditions, and they live in relative harmony. But like I mentioned before, this is not really necessarily the most accurate. It's good. It's a good basic starting point, but it's not necessarily the most accurate representation of real life, of the real life. So let's start adding some more complicated additions to this. So one of the additions we can add is something called the alley effect. And the alley effect is basically a biological phenomenon that correlates population size with the growth rate. So basically, it's basically that if a population has a really small density, it is difficult for them to survive and reproduce because it's more challenging for them to interact with similar members of the species. So this can take effect in 2 ways. One of the ways is there's 2 types of alley effects. One is called the strong alley effect, and one is called the weak alley effect. The weak alley effect just basically says that if the population density is below a certain threshold, the growth rate is lower. It's still positive, but it's lower than normal. The strong alley effect is basically saying that if the population density is below a certain threshold, the growth rate is actually negative. So there's a decay, and eventually the species will go into extinction. Mathematically, this is represented by modifying the growth function, and the growth function is, if we look in the simple Lotka-Volterra model, basically this can be substituted usually just in this component when it's multiplied by the A. That's the growth function of the prey, sorry. So that can be substituted by this equation. And this equation is basically comprised of the intrinsic growth rate of the prey species, and then in times 1 minus x over the carrying capacity of the system. And this is all multiplied by x minus m. And x minus m is the effect of the alley effect. So the alley effect is basically the m, such that if m is greater than 0, you have a weak alley effect, as in the growth rate will still be positive. If you have a strong alley effect, you will the M will be negative, and yeah, sorry. Strong alley effect is when M is positive, my bad. And the weak alley effect is when M is negative. Basically, the way this works out is basically if M is large enough, you will basically have the effect of if X is below the certain carrying capacity, you will have a negative growth rate. And then basically we can add this to the Lotka-Volterra equation by substituting our A, which was our growth rate in the most basic equation, but it's actually the growth function in the more complex equation with what we have here. We still maintain our negative rate from the predator interaction, and then the predator equation stays the same. So to see some of the alley effects in action, we're going to start with these parameters. This is going to be the weak alley effect because M is negative. Let's run it. Okay, so it just auto starts with the parameters that we wanted, but it won't print for some reason unless I modify it. Okay, so this is also an issue I've been running into. I don't know how to delete the previous slot whenever I change it. I don't know if someone has a solution to this, but it basically will just keep
I was
stacking
having the
up.
same issue. I was having the same issue, but I think so the way I did it, I separate the cell where you input the parameters and the cell that you execute the graphs. They're different cells.
I
So
think
you have to
when you
click
do a
two
simple
times.
print command after the cells, that might fix it. I have not tried, but I think that might fix it. Just an empty print statement.
Okay, that makes sense. I'll try that then, I guess, after. But for now, I guess we'll have to suck it up and look through the multiple plots. So here,
So the problem is, maybe to give you background, the problem is that in Python, you say that you want to print this plot, but then Jupyter as well prints it. That's the problem. So that's why it's get twice printed. Yeah.
Okay, so if we recall from our equation, it would be more useful if this had disappeared, but if we recall from our equation, we can look at, let's take a look at the carrying capacity. And the alley effect is a percentage of the carrying capacity. So if we change the carrying capacity, we should have a different point at which the alley effect, I guess, takes place. So if we change, yeah, so here we see the alley effect is kind of changing. If I increase K again, actually lessening the alley effect. Bring K back to its normal value, and let's change, well, R is pretty, I guess we can think of that one pretty easily. R is just the growth rate. So if we modify the growth rate, we should, let me just restart this. Yeah, so this is the difference between changing R. If we change R, this is the modified R. We basically have, this is sort of because, okay, this is the original R and this is the new R. Okay, let's take a look. This is the original R with R equals 2. This is the initial growth rate. This is the new growth rate. Here we can basically see the alley effect. So I guess initially we're looking at the alley effect. We can basically see that if the prey population is at a certain point, it basically will drop and approach this fixed point over here. In comparison, if we change the growth rate with this, we're basically just moving the wait, maybe I'm not even understanding this myself. Seems like just the slope of this line is what's changing when you change the growth rate. And if we change P and C are predator related parameters. So if we change P, we basically just see a higher predator concentration. That one makes sense because this is the effectiveness of predators on at eating food. So if we increase the predator effectiveness of eating food, their population equilibrium will increase. And if we increase the predator, let's restart this again. If we increase the predator decay rate, we will see that the equilibrium for the predators is lower than what we started with. That also makes sense because that means that the predators are dying faster. Okay, so now let's take a look at the next one, which is the fear effect. And basically the fear effect is a scenario where predators, basically it's a way to simulate how prey will behave in fear of predators. So this presumes that prey are aware of the presence of predators and they feel fear and act accordingly. And this makes them try to avoid predation, which can increase the short-term survival of the prey, but oftentimes causes long-term consequences. So this is the new formula that we have. This is in combination with the alley effect. So these middle two terms are the alley effect, and it's basically just been simplified with less parameters. So it's just, if you combine all the parameters and multiply the whole alley effect out, you can get something with just a simplified parameter A and a simplified parameter D. But if we take a look, these two components are what we started with the initial Terra model. And basically what we're doing is we're modifying the classical predator-prey model to incorporate this fear function. And this fear function affects the growth rate of the prey. And we are also substituting in the relationship between predators and prey with a modified growth function. And the simplest growth function is just x times p, which is what we had before. But you can also modify this to have more complicated growth functions, like x times p over 1 plus qx. This is another possible growth function. For our case, we will still be using just the xp, just because it's the simpler one. And basically, this fear effect represents the level of fear that the prey have towards the predator. And this can be simulated by basically any function that meets these parameters, which is basically just a decaying function. So basically, as s approaches infinity, y goes to 0, f of sy goes to 0, and as y approaches infinity, f of sy goes to 0. We are going to be using a specific f of sy. We're going to be using this as our f of sy, which is 1 over 1 plus s times y. This is just the simplest sphere function we can use. And we are basically going to be plotting that. Let me root on Excel. Okay, so these are the simplest values we can use to, I guess, get a visualization. We're going to start with these. Okay, so here we see if we use this, we basically get a scenario where the prey population and the predator, the prey population, if it's below a certain point or a certain line, it will approach that line. If it is above that line, it will decay to that line, and then it will stay basically on this line until the predator population hits 0. So this is an example of when the predator when the fear effects causes the predators to population to go extinct. Yes, Pedro?
It doesn't seem like it's going to 0 because if you see the arrows on the left, they're
Here, yeah.
pointing to the right.
Yeah, that's a good point. So this is the example they gave for us for when the predator population goes extinct. And I totally agree with you. It doesn't actually look like it's going extinct here. But that's just, I'll look further into this to make sure that I plugged in all the numbers correctly, but I double checked it like 3 times. So I think I did. I think when they graphed this on the paper, basically, it was just a blown up version. So it looked like it went to 0.
It probably depends on the parameters. Probably both can happen, I think.
Yeah. I'll take a look at the
Doesn't-
parameters again, because these were the ones that they provided for us.
Mm.
I'll just make sure.
That's a problem.
Yeah, okay. So we can also modify these parameters. I didn't write them down here actually just yet. But we can also modify these parameters so that we can get a scenario where just the prey goes extinct, or both goes extinct. We can change this so let's just try increasing the fear effect and see what happens. Yeah, so here, if the prey are more scared, there's an exaggerated version of this where basically it more rapidly approaches the predator extinction. And this makes sense because if the prey are more actively avoiding the predators, it will take less time for the predators to go extinct. If they are even less scared, let's see what happens. Yeah, just what we would think it would take longer basically for the predators to go extinct and even it seems that the predators don't necessarily even go extinct because the prey aren't even scared enough. There's actually seems to be a fixed point here, a stable fixed point. And then you can also just, I guess, play around with the rest of these values, try and modify it. So A and D are related to the alley effect. So if we modify these, we will be modifying the alley effect. Let's try modifying A. It looks like the ecosystem is in more harmony as there's greater predators and still prey still seem to approach a stable fixed point. This looks like a good ecosystem to me. Okay, so that's this one actually. So yeah, here we see, this is actually even better. we're seeing the alley effect in action, which is if the prey are below a certain threshold, we have a change in behavior, basically. Because that's what the alley effect is basically if there's a certain threshold of the carrying capacity, there's a change in the dynamics, which is kind of what we see here at this point. We are seeing a different dynamic.
Can I
as
see if
we're-
I understand? The alley effect is the slope of the curve when y is close to 0. Is that
when
right?
x is closer to 0.
So I already forgot who is the predator, who is the prey.
X is the prey, y is the predator.
X is the prey. Okay. Okay.
Yeah. So
Yeah,
basically,
So when x is close to 0, the slope is like pretty 90 degrees, which means it's growing very, very slowly. Yeah, okay.
yes,
Got it.
correct. It's growing very slowly here. Sorry, very quickly. But we see, more importantly, we see basically the point of the alley effect is that at a certain threshold of low prey concentration, there will be a change in basically behavior, or a change in the growth rate, which is kind of what we see here. So the last one that I wanted to do, I didn't have enough time to implement this into code basically, but we were going to do cannibalism. And cannibalism is even easier than the alley effect and the fear effect because it is just taking the original Lotka-Volterra model, which is this, and then adding this cannibalism function, which is C1 of X or C2 of Y. And hopefully this will be done by next week. But basically, there are specific functions that they have listed in the paper that you can do for C1 of X and C1 of Y that are dependent on its own population, as you can see, because it's C1 of X and C1 of Y. So it's just another food source for itself, which is kind of morbid, but I mean, it's a behavior that we see in the real world. So they decided to model it. That's basically what we're looking at right now. If there's any more questions, I'll answer them.
I have a few questions and commentaries. So first of all, in your introduction,
Yes.
since you will publish it or something like that, I think, I mean, people could disagree, but if you read this again, to create modeling equations for us to better be able to understand the mathematical relationship that predators and prey had in each other's population. So the mathematical relationship between predators and prey, this is your model.
Yes.
So this is the model. So basically, the way you phrased it, it seems like we are building a model to understand the model.
Yeah, I see what you're saying.
Okay, yeah. Next, I would like to see the m greater than 0 case, which you didn't show in the basic alley
yes.
effect.
We can do that. I have, I wrote
Yeah,
down some good values for this one. We can do that right now if you'd like.
I think it's just putting like +2 instead of -2. Would it work?
It would, that technically is what we want, but the problem is the parameters will all have to change so that we get a graph that I guess fits the field of vision that we want. We can just
Okay.
try that. We can do +2. I don't think it will look good though.
Mhm. It's all interconnected at this point, right? So
Yeah.
it's kind of a mess, I understand.
Yeah, we're just getting some strange behavior. We can just try changing everything to one of their recommended ones though quickly.
One thing you can do is you can close a cell. So go up in the three dots, right corner, the three dots in the cell. Yeah, I think it's add a form. Add a form, the last option. And then there is an arrow. Yeah, close the arrow in the left in the cell. Yes.
Oh, okay, awesome. That's very
Yeah,
valuable.
that's it. Uh-huh. You can still access your code clicking show code, but looks much, much better and you can use it as an interface.
Yes.
What is the trigger to generate a graph?
Any change in value.
Ah, okay. So it generates so the last graph is the
It should be the last
corresponding
one.
one.
Yeah. So here we see the
So
last
X
one.
goes to 0. Increase M a little bit. No, it doesn't matter. We should zoom in between 0 and 0.5 actually
Yep.
to see this graph. Can you do that?
I would have to change the code.
Yeah, don't.
Yeah.
That doesn't matter. So
But we can change some of the stuff. We can change the growth rate so that we can increase the size, make it higher.
yeah,
The predator relationship, which is P.
but increasing P also decreases X, right?
That would also decrease. So yeah, that's why we have the 0. So that doesn't even work. We can decrease C, but it's already so low. So it probably wouldn't even matter. But yeah, I guess I can. It's going to be a little more challenging though, to if I want to code a graph
Set
size that changes.
you set the limits, X and Y limits as input
Yep.
parameters. So
Oh yeah, I can do that. You're right.
yeah, this should be easy, right? And then you
Yeah.
just, I mean, your model is probably inside a function, is it?
Yeah, it is.
Yeah, so just create another input and that's
Yeah.
it.
Okay, that should work too then. Yeah. But basically here we are seeing that you do get there if the population is above a certain point, you get complete extinction basically.
Yeah, nice.
All right, any more questions?
I forgot the other ones. So yeah.
I think this is the kind of thing, I mean, these are good comments. I have a few. I agree with Pedro. It would be nice to be able to change the field of view of your plot so that depending on what you need, you can change the C-bar. Might also be helpful to have some time series plotted for
Yeah,
different examples.
that's a good idea too.
And something doesn't quite work because for a lay effect, is the lay effect on the predator or on the prey? It's on the prey.
It should be on the prey, yeah.
So
Which, yeah, you're right. This is actually
in that
confusing.
case, so X is the prey. And if there's an allay effect, then when the population of prey goes below some threshold, it should go to 0.
Yeah, that you're right.
And
This
I'm,
is the
but
opposite
I'm
of
not,
what we
and
were
I'm
doing.
not seeing that happening. So I'm confused a little bit about why the effect is working the way it is.
Yeah,
But
I'll look more into this also then.
again, that's one reason to have a sort of an in-depth session with one other person, so you can do some debugging and cleanup.
Yep.
Okay, any other general questions for today?
Yeah, I mean, I have general comments once, but we have as well together session, I guess. So what I wrote down is would be good if you maybe able to access not only with X and Y, but as well say Ray and predictive. And then you have kind of like these two different stages, like you had the strong and the weak alley effect. And I wondered if could do something continuous because it goes from negative from a positive value to a negative value. And somehow these are just two snapshots, but maybe it could be interesting to see how this changes when this will be continuous. And
I could try and make a slider that goes between these two
yeah,
and
cool idea.
modifies everything linearly between these two. I could try that.
And then one more idea had you could combine all of these effects so that you have a effect, field effect, and cannibalism in one.
Yes.
Then when I read the plots right, there is always one fixed point. Is this correct, or is it sometimes as well so that you have two fixed? No, you never get two fixed points and bifurcation or something. It's always one fixed point.
Yeah, we're basically, it seems like every scenario
Yeah,
is we're getting is
there's not even a fixed point. It's basically.
yeah.
That are all my notes I wrote down so far. Yeah. But nice exploration of the model. Thank you.
Thank you.
Okay, any other questions from William or Alex? Okay, William, go ahead.
Can you hear me? Can you hear me 3 years?
Yep.
I thought it all made sense. Adding it was very it made sense to me how you started with the basic equation and added a more complex like system or parts to the equation. I was just hung up, or not really hung up, but I think that being able to change every single parameter is a lot to like a lot at once. That's just for me. I don't know. But yeah, otherwise it's good.
Yeah, that's true. Maybe I'll try adding
Okay, great.
snippets in between every thing so that you can, I guess, know what each thing does when you change it. So that maybe it's not just as so overwhelming just to see all of them at once.
Or you might have some exercises where you say, try this value, try this value, try this
Yeah.
value. And then maybe give the person a little bit of what they might expect to see.
Yeah, because the free reign, I mean, it's like what? But like, I could put something in there. I could completely step up. That's not the whole thing.
Okay, I'll do that. I'll add exercise, like guiding exercises.
Ibrahim,
Yep.
maybe one thing that would make it would make your application like wonderful, but so it's a suggestion. It's something if I looked at and played with it, I would say, wow, this is brilliant. Which is to have some checkboxes. It's called a radio button where you click and it checks. And then you can check, for example, so you have the base model, and then you check fear and
Mhm.
check alley, check cannibalism, but uncheck fear. And then when the person select each kind of behavior, you run the corresponding model to the
Yes.
selection you made. This would be brilliant, but it's work, so it's not even a suggestion. It's like I wondered.
I can try it. I'll try it.
No, those here.
Okay, great. Well, those are great points. Again, if people have time to type their comments, that would be appreciated. Our next presentation is supposed to be from William.
Okay, so I worked on, how do I get rid of this? Sorry. I worked on P53 and DNA damage, and I will go into more in-depth biological background next. How do I change that? Okay, cool. So essentially, in all of our cells, DNA is constantly under stress from various environmental factors, as you can see in the diagram, from UV rays, like smoke, anything chemical, biological, etc. And cells have developed complex mechanisms to combat this stress. I studied the P53 protein, which is essential for telling the cell, oh, when does it kill itself, or when does apoptosis occur, or when do we repair this DNA damage, or when do we put the cell into cell arrest, or when do we put the cell into senescence, I think is what it's called. And so essentially P53, the protein, lives in a system that is regulated by MDM2. MDM2 is another protein that suppresses P53. So in a healthy cell, P53 is low to start. Can you see my cursor? Yeah, I guess it doesn't really. A whiteboard would be, no, whatever. So P53 is low in a healthy cell, and MDM2 is relatively high. And when DNA damage occurs, MDM2 degrades, and therefore causing P53 to increase. And then this P53 increase causes MDM2 to be produced because it's a positive negative feedback loop. It's an auto regulatory feedback loop, sort of like this diagram. Okay, one last biological background slide. Okay. Levels. Oh, and an important thing that researchers have discovered semi-recently is that the levels of P53 and MDM2 rise and fall in pulses, or like limited oscillations, if the DNA damage is strong, like relatively strong. And they sort of wanted to know why these pulses occurred, what was like what caused these pulses, and yeah. Okay. What'd you say? MDM2 is another protein. MDM2 is a protein that, what'd you say?
Yeah, it's okay. Of course, it's not protein.
Oh, it's not?
Yeah, it's what the function or so. That's why. I mean, it's a pathway, right? You have P53, so it's MDM2 built somewhere with the same pathway, and
Oh,
you'll have as well some functionality.
okay.
It's okay. It's not.
Okay. So it's not so what is it then? It's a pathway?
I don't know. I would have to look it up myself.
So then what are you telling me then?
The idea is just that these proteins work together, right?
Yeah.
So, or they are at least correlated.
Yes.
So on P53, you really nicely described what it was. So it decides if it kills the damage or if it kills the cell. And then the two, if you Google it, I'm sure there's a Wikipedia page. It will as well have some
Okay.
something that is the big word.
Got it.
And then maybe it's interesting because it will maybe as well be DNA repair
Yeah.
pathway. Maybe not, maybe they cover something different.
Yeah.
For I don't know, skin color or so. I would say so, right?
Yeah,
But
yeah.
it could be right, it can be anything, right? So it might be interesting to know what it is if you only talk about two proteins.
Got it. Okay. Noted. Thank you. Okay. So I wanted to investigate this system and make it make an interactive sort of notebook for the user to be able to familiarize themselves with this system, because it's important as P53 and the P53 MDM2 autoregulation sort of system helps stop tumors and DNA damage and et cetera. Okay, so the model already exists. I sort of took the model and adapted it a little bit, added some things from a paper. And I will now review the ODEs, which is on the next slide, and go over how they simulated DNA damage because I found it interesting. Oh, interesting. Okay. Apologize for the like granularity of the image, but so these three equations are what constitute the changes in P53, MDM2 in the cytoplasm and MDM2 in the nucleus. Okay, I got that right. So we can see P53. I'm just going to go over maybe one equation. This is the basal rate of transcription of P53. This is the rate of P53 when in a healthy cell, in a normal cell. And this is our, wait, I'm blanking. Is this not our Hill function?
It is. Promotion.
Yes. And this is our degradation term. This is with the degradation constant in P53. This is similar to equations we've seen in class. And so I took these equations. Oh, and notice that MDM2 in the cytoplasm, and there's MDM2 in the nucleus, because these two compartments, the cytoplasm and nucleus, affect the system in different ways. So you have to separate them. And so I essentially took these equations and changed some of the parameters, which I will go into later. DNA damage, they simulated DNA damage by using this Goldbeater-Koshland function, which is very hard to understand. I tried to read the paper, but it's essentially a function that is used when there are feedback loop, essentially a function that scientists use to simulate feedback loops that lead to like oscillations. And also they used just a binary step function. If x is less than 0, then it's 0. If x is greater than 0, it's 1. This is essentially saying if there's DNA damage, then like it's on. If it's not, then there's none. And this is, so then we get down to the damage degradation rate constants. And so the change in DNA damage is the repair rate times the Heaviside function of DNA damage. And how these constants affect the model is that KD2 is the degradation rate of MDM2. If we go back, so KD2 is here, and KD53 is the degradation rate of the constant P53, and that is here. So DNA damage affects all of these constants, or DNA damage, the change in DNA damage is here, and DNA damage affects this constant, and thereby affects the other equations, since they're all related. Okay, so I essentially just plotted the initial model with all the initial parameters. The parameters are the steady state values that are calculated from solving the ODEs by hand. And we can sort of see what we expect. DNA damage comes in at time equals 50. MDM2 in the nucleus begins to drop quickly due to the DNA damage, and this results in P53 increase. This P53 increase in blue will result in MDM2 in the nucleus to be produced, and this production of MDM2 in the nucleus this production of MDM2 in the cytoplasm will eventually peak and start to decrease as it starts to sift into the nucleus. And that is why, so the orange is increasing and the green begins to increase because the MDM2 in the cytoplasm is increasing at a very fast rate, and and it is also sifting into the nucleus. I don't know if sifting is the correct term, but and then they eventually end up coming back to their steady states. So that is one pulse. Oops. So that is one sort of pulse. This is when the DNA damage is relatively low. This is one pulse of the system. Okay. So now I wanted to explore DNA damage and allow the user to see how DNA damage affects the model and the graphs, and if we can see pulses or separations and whatnot. So I essentially created a slider that you can't, I mean, it's presentation mode, you can't really see it in action, but here I made the slider maximum value around 4, where the first pulsations come. So there are 2 pulses here, and this correlated to high damage. I essentially identified a parameter within the model that linearly influenced the amount of DNA damage, and I'm just increasing or decreasing it. And then here, while we decrease the DNA damage, the system does not react as forcefully. So the amount of MDM2 in the nucleus does not have to decrease that much in response to a lesser DNA damage. And I found these pulses interesting. And so I wanted to see, okay, how much could we push this? So I turned the parameter up to 6, plotted it for a longer period of time, and saw 4 pulses. The function of these pulses it's sort of debated upon. There's no, I think the function is that the system is trying to repair this DNA damage, and it takes multiple pulses in order to do so. And scientists have not found a correlation between the amount of pulses and the decision that the cell makes yet, which I kind of want to look into in the future, but I don't know how. And moving on. So I tried to plot the bifurcation diagram, and I think I can try harder. It was oddly hard with the model that I had. And so this is a bifurcation diagram from another paper that I picked up. And so this shows that once we hit, so DNA damage is on this axis, P53 is here, and then time is here. So it's sort of a 3D bifurcation. So once we hit this amount of DNA damage, then we will stop we'll go away from the steady state, and we will either have a maximum or a minimum, and we will be oscillating between the two. Okay. Now I wanted to just see what other parameters I could explore within the model. So I chose the basal rate of transcription of P53, which was here. And I wanted to figure out, okay, I mean, I sort of expect the system to, quicken the production of P53, since the basal rate of P53 is increasing, and and it does. So when the basal rate, the change in basal rate is 0, then we just have our normal function. But or our normal graph. But when we increase the Basel rate by 0.4 here, the graph shifts to the left. When I mean the graph, I mean all of the pulse essentially shifts to the left and is is over faster with in time. I then adjust. I just want to explore P53 production a bit more. So I adjust KS53 by multiplying it by a scalar, and this is sort of what we expect when we multiply KS53, or when we make KS353 smaller. Then the lines do not overlap, meaning that less P53 is being produced, and when we increase it, the lines begin to overlap and shift to the left again. Makes sense. Next, I plotted phase plots or phase diagrams of the three variables altogether. So with P53 on the x-axis and MDM2 on the y-axis, we can see that all states seem to converge around this point, which is approximately their steady state. And I found what was interesting here was that the lines are the lines are more horizontal than vertical, and this shows that P53. This shows that in each state, the state wants to get back to P53's steady state faster than at the MDM2 steady state. And moving on to MDM2 in the cytoplasm and P53 on the x-axis, we see something interesting here. So there is the same steady state point here, but now we can see that the arrows diverge and go to infinity if they start at some state value. And this was interesting to me. I think this occurs because the parameters are off. I don't know what else would be the problem. This does not signify any oscillations because some of these parameters I tested, and for example, if I just test 0.2, 0.6 or something, and just keep the MDM2 and the nucleus the same, it does not cause an oscillation.
Question on that one. If you actually plot the time series, do they really go to infinity, or is it just outside of the scale of 0 to 1?
I'm not sure.
I'd check. It may be like what we saw with Ibraheem, where we need to change the size of our box.
I changed this. I did not plot the time series, but I changed the phase diagram to what, like 10, and it was still going. So
it's still okay. So it may well diverge. But,
yeah.
I just sometimes those things have a fixed point, but it's large, and so you don't see it when you're looking at the lower value fixed points.
Lastly, I plotted MDM2 against in the cytoplasm versus the nucleus, and this was a pretty expected relationship since they're linearly related or correlated, and they both converge at their respective steady states. And the lines are more horizontal than vertical, meaning that MDM2 in the cytoplasm quickly converges to its steady state faster than that MDM2 in the nucleus. And that's all I got to so far. And yeah.
Did you want to show your notebook too, or was that?
Can I show this?
I mean, it occurs to me seeing this, which is quite nice, that one of these things, when you do these phase plots, it really would be helpful if we actually had the positions of the fixed points. And so one way to do that would be to run the time series for a long time and save the final values. And then you could draw a dot on top of the phase plot where the fixed points are.
Yeah.
So that it's clearer where the fixed points are. Because in some of these, if you don't change the density of the lines in those phase plots, it actually is pretty hard to figure out. Everything gets crunched together, and then it's actually rather hard to figure out exactly where that fixed point is. And if one were ambitious, you could even do what we did in class one day, which is run things backwards in time and find the unstable fixed points too, and put a dot there. So I probably need to write some code for myself to find stable and unstable fixed points and overlay the phase plot. It's not very pretty. I mean, it's sort of kludgy to do it that way, but I think it would make the diagrams much clearer if we did that. Another thing that I was thinking of just generically is do remember to include references.
Yeah, I have. Oh,
and something that's
Yes.
nice, and there you have them there. But in your notebooks, of course, you can have a link to the reference.
yeah,
So the user can click on the link and then be taken to the Wikipedia page or wherever you have a description of what P53 is or what
well, yeah, I was wondering.
MDM2 is.
Well, I was gonna ask, do you care about the format at all or anything like that, or?
No, although it's useful if you can link to a viewable
Yeah,
version
that
of
makes
the
more
paper.
sense. Yeah.
So, for example, if you have a DOI, that can be written as HTTP :
Mhm.
and then you have, in principle, a click that will take you to the original page.
Yeah. Okay. So, I mean, here's sort of my notebook. I mean, maybe I could run it.
Great.
Um,
And we need to add those magic lines of Elmer's to suppress all of the dialogue at the beginning.
yeah. Wait. How do you? Is it not that, or is there more?
It was
Oh, maybe.
percentage.
There's a single decorator that tells it not to output the
Percentage capture.
Capture this.
Yes, but on the top.
at the beginning,
Beginning first.
and
First line?
it hides it.
Yeah, maybe.
But-
Or after line 2 would be fine.
Okay.
Yeah, there. That hides. Of course, then if you have an error, you won't see what it is. But at least that way, it hides all those warnings that are not very interesting.
Got it. Okay, so I just load the stuff or import all the
Mhm.
libraries. Have some biological background,
Yeah.
a lot of stuff. This took a little bit of time writing the equations in
Yeah.
markdown format. I tried to explain them here.
Right.
So someone who doesn't know much can try to understand.
I think in your verbal presentation, you did a great job of saying term by term what's going on. You
Yeah.
said this is the source, this is the basal metabolism term, this is the decay term,
Yeah.
this is the regulation, the production of P53 by MDM2 and so on.
Okay.
That was very helpful. One thing that might be useful would be to actually plot also the level of DNA damage versus time.
Okay.
And maybe also to show
Oh,
you have this complicated function KD53, which has this rather complicated form.
yeah.
It might be nice to have a plot of that, not maybe for everything, but just to show what the shape of that is.
Yeah,
In
yeah,
other
yeah.
words, is that increasing or decreasing as P53 increases?
Yeah, I see what you're saying.
Well, because I think the shape of that function is probably pretty critical to what's going on to get these oscillations.
Yeah, I think, okay.
I'm very glad we did FitzHugh-Nagumo last week, because we had an excitable system, And here you're seeing another excitable system, right? You see a situation where if you don't do anything, it sits at equilibrium, but if you push it and you push it a little bit, it comes back. If you push it a lot, it spikes and then comes back.
Yeah.
And that's a classical excitable system.
Yeah.
Goldbetter was very, very famous for studying those, which is why there's a Goldbetter function.
Okay, that's what yeah. Okay, so I use Pedro's method, or actually Hayden told me to do this. So you can put this at title or hashtag at title, and then a label, and that creates a title, and it's sort of what Pedro did.
Great.
But so run the model. This is the model. This is just the basic. I sort of explain just in short stuff. I introduced DNA damage. I explained changing. I explained the constants again, and then I create this sort of interactive DNA damage slider, which is kind of cool.
So that's the amplitude of DNA damage, but the DNA damage is only done once at 50.
Yes.
So I think it wasn't so clear maybe from your verbal presentation. What's interesting is you damage it once, but you get, as you say, you get multiple spikes of response.
Yeah. So,
and that's actually a bit surprising. So you trigger it once, but then you get neurons will do this too. They're called bursts, where you trigger a single action potential, and then it overcompensates, and it does another one and another one. We did the two cases in class where you either have constant firing or only a single pulse, but you can design neural systems where you get one pulse and then you get maybe 10 pulses or 20 pulses, and then it turns off again. And so I think the idea was clear later on, but I might say that from the beginning that this actually is behaving in this way. You're going to get these, and that's really neat. They get pulses.
Yeah. Okay. So do you think I could overlay? Do you think if I could overlay DNA damage, like it would be a shoot up right here, and in the background, it would be probably it's a linear decrease.
Maybe an exponential decrease. Yeah. I don't know. So you can use it's not very fun with Matplotlib. You can do layouts where you have multiple graphs in a frame.
Mm-hmm.
So you have like one plot here and another plot underneath it.
Okay.
If you look in Herbert's demos, he shows how to do those layouts.
Okay.
They're a little bit of a pain to set up. Once you figured out one, they're not so bad.
Okay.
I tend not to use them in class because the things are small enough, and then if you have lots of plots in a screen share, it's the other thing that might be rather interesting in this plot, it would be a little bit more work perhaps, would be how many spikes do you get as a function of the DNA damage?
Yeah.
Right. Yeah. You could have a plot where the x-axis was the damage amount, the damage level, and the y-axis was, if it's very small, you get no spikes. If it's within some range, you get one spike, two spikes, three spikes. That could be, I don't know how you feel about that, Elmer, but to me, I think that could be quite interesting. How many spikes do you get?
Why are you asking?
Well, it just seemed like the thing is something that yours would be your style. That's why I thought maybe it would feel
I could do that. That just reminds me of the thing we did in class of right, counting the oscillation or
finding the peaks.
Yeah, the peaks. Okay, I could do that. So I talk about some observations, and then I sort of just plot it again. So maybe I can make this interactive here, and examine there are four pulses here. Bifurcation diagram. I tried to plot it. I think it's because the model. I'm unsure actually. I tried, and the problem is the model.
Yeah, well, the bifurcations in this case are a little complicated because we talked about bifurcations where steady states change.
Yeah.
But in this case, you still have the same steady state.
Yeah.
What's happening is whether you're excitable or not.
Yeah, I don't know how to.
And so, if you have something that counts how many spikes you have,
then I guess that's
as a function of a parameter. That is the
okay.
bifurcation
Okay.
diagram.
Okay.
So if the question is the stability versus instability of a single steady state, it's a little bit different. Or
Okay.
again, in the class, I'm afraid we tried to talk a little bit about stability, but we didn't get through all the possibilities of stability, which is sort of a big topic.
Oh yeah, this was my attempt. It was poor. So
I have a
then I
question.
went into just trying to figure out what else I can look at. So here I can change the Basel rate, and it sort of just shifts the whole graph, which I think we've seen in class. Then I changed the
The decay rate.
No. I changed another rate. I should have the equation right here. I don't know. Here, I changed this production
The
rate.
level of activation.
Yeah, the
by
level of
MD2.
activation. And if that one, it's the default, and then
Yeah, that's pretty neat. I mean, so there you really see that if you go down again, you really do see a bifurcation, because keep going slowly. From there to the next one, you go from sub-threshold to above threshold.
Yeah.
For the damage.
Yeah.
Right? You go from no
So,
pulse to
William-
1 pulse.
Yeah, I see. Yeah.
And then
William,
presumably there's some other one where you're going to potentially go to more pulse.
Yep.
William, is
So
the,
that
like,
really is a bifurcation. Unfortunately, it's not one of the classes of bifurcation that we talked about in the past 2 weeks.
Okay.
If you're interested in that, the textbook, the Murray Mathematical Biology textbook does do the analysis of that problem. But that's probably a lot to ask for in the last week of the semester. But if you're interested, that particular bifurcation type is treated.
Before we move on,
Okay, so
can you hear me?
then I was sort of trying to figure out what else I can do. But a lot of the parameters I was messing with were hard to understand, like hard for me to understand what was happening. I guess this does not work, but I don't know.
Well, there
And
was a question in the chat: is the only relationship between nucleic and cytoplasmic MDM2 diffusion? And yes, I'm sorry, I have to mute the people who are online, otherwise we'll get feedback in the room. That's the limitation of having multiple. Actually, I guess what we could do is if William turns the speaker on in his computer, that it'll probably work. But people can put the questions in the chat, and I'm happy to relay them.
yeah, the relationship between nucleic and cytoplasmic MDM2 is yes, diffusion. If diffusion, I assume diffusion is going from one.
Right. So K out and K in are the transport rates.
Yeah.
And so that's something you could change. You could change the K in a little bit, and that'll mean that for a given amount of material in the cytoplasm, the balance between how much is in the cytoplasm and how much is in the nucleus will change.
Got it.
But
What'd you say? This is how it came. Yeah.
so no, somebody asked a question: is total MDM2 constant? No, there is production and decay of it.
Yeah. Right.
But it's all produced in the cytoplasm, because all
Yes.
protein production happens in the cytoplasm. So then it has to be transported to the nucleus.
I don't think there's a difference. Like you're saying this versus
Yes,
that. Yeah, I don't think. I mean, I don't think. Yeah, I don't think there's anything. Yeah. Okay, keep going, and then I just was going to plot face plots. Yeah, let's see if it works. Yeah.
So,
So what I found interesting here was the like direction of arrows, or not, yeah, sort of the direction and whether they're horizontal or vertical. And so I just plot MDM2 versus P53, P53 and MDM2 in the cytoplasm, and the two MDM2 in the cytoplasm and nucleus. And this is where I stopped.
Okay, can we come back to the one where you go back up one to this one here, where you say that the values run away?
Yeah,
What are the parameter values where that happens? Because when you plot your time series, you should see the values go to infinity.
Yeah, okay, maybe should I just plot the time series here?
That wouldn't hurt. I don't want to take too much time, but I think that's quite interesting. People have done a lot of interesting work.
So let's do this.
So plot.clear doesn't work I'm asking while you're doing, you type.
Okay.
Elmer and Pedro, does plot.clear not work?
So the issue is
You got plot.show, but I don't know if plot.clear works or not when you want to erase an image.
But it should, because it's not complete.
Right. It should be plot.clear and then open close parenthesis should erase the plot.
can you hear me?
Yes.
So the issue is the creation of the graph is done inside a function.
But the other ones can't, because I have my headphones on. So Pedro would like to say something.
Pedro, go ahead
Yeah.
and put it in the chat.
Okay.
Here, Pedro, go ahead.
Can I talk? Are you hear me?
Yes, go ahead.
So the issue is that the creation of the plot is done inside a function. And this function is in another cell. You cannot like clear a graph in another cell using a function defined in another cell. It doesn't work. And if you do that in the current cell, you clear everything, including the widgets.
Ah, okay.
So I couldn't find a solution to it.
Yeah, I'm actually confused because it's not.
So I don't think we can necessarily solve this here
Yeah,
today,
yeah.
but I would say why not try looking into that a little bit in the next few days. As I say, I want to make sure we get through everybody today, and I did want to show them at least a little bit about 2D bifurcations before the end of the. I think that's great. Any more comments for William?
Yeah. I'm impressed in how much progress he made in continuous flashing.
Thanks.
Asking what all they means and now it's a space lots and talking about biology.
Great.
Hello? Can you hear me? Hello.
There was a good question, which is, if you can share with the class your links to your notebooks so people can take a look. That would be great. So let's see. So Pedro, I think you're next.
Hey.
Actually, let's take a 2-minute break, 5-minute break, and we'll come back to Pedro, okay?
Can you hear me?
I'll let you set up. I'll be back.
Hello.
But I think you have to speak, John.
Wait, Pedro, what are your suggestions?
they say they have donuts upstairs.
Try it, steal some, go ahead.
I'm good.
Okay. So, I think you should put like the key experimental data they provide if they do, because
Uh, wait, I was echoing. Are you saying experimental? Like, actually, sorry, I interrupted you. I do not fully understand. You can like they came up with initial parameters for the model. I included that in the notebook.
yeah, but no. This would be the fit parameters or whatever. What would be interesting to see is what they measured in the experiments. For example, time series, I don't know, whatever they have of observations. Do they have that on their paper? Or it's just a model, mathematical model?
It's just a model. I think
Okay.
they may cite papers. They cite a lot of papers. They might cite papers that actually run the experiments, but it's just a model for this paper.
Okay, never mind. I mean, it's interesting to see the experimental counterpart, but if they don't provide any, then why bother?
I guess, yeah.
Great, did you find the donut?
No, they didn't.
Ibraheem said, were you able to get the whole model from online SPML? Yeah.
Then were also the modifications you made, did they explore those in the paper also, or did you just like decide to explore those on your own?
No, I just did that.
Okay, nice.
I thought it took me a while to find ones that would work. But yeah, I just decided to modify some things that I thought was interesting. And yeah, you can go.
Go ahead. So also this pathway that you're describing, are there like other similar pathways for other, I don't know, repair mechanisms or just other mechanisms?
I don't know. I can find that out. That might be interesting to add. Okay, cool.
Okay, I mean, I don't mean to stop the discussion if people are having a good time. Or shall we move on to Pedro now, or do you want to keep talking about William's project a little more?
Good.
Okay. Okay, great. Okay, Pedro, take it away.
So, my project is the replication attempt so far of the Nipah virus immune antagonism strategy. So, this is my Colab notebook. There's a disclaimer. I'm not sure if it really reproduces the paper results, and I don't think I will be able to check that because they do some mysterious normalization of the results. So it's a little bit unclear what's going on. But the shapes, the graph shapes, they look similar and they behave the way they should behave. So I am like 40% confident. First of all, I give it an introduction. So basically, Nipah virus is a very deadly virus. So if it infects you, it has at least 40% chance of killing you, and it goes up to 75% chance. it causes encephalitis, I don't know how to pronounce it, which is brain inflammation. And if you don't get killed by the virus, you will be left with um how do you say?
It leaves you with brain damage, basically. Long-term after effects, yes.
Okay. Do you use this word here, sequelae?
Technically, yes. If you're talking
Okay.
about medicine, yes, a doctor would say that.
Oh, okay.
I mean,
I will
there's a
probably
version of that word that's very common, which is a sequel, as in a sequel for a movie.
Oh,
The
sequel.
same word.
Mhm. Okay.
Sequelae is just the plural of sequel.
Okay. Sequel. Oh, interesting. Yeah. So you can have brain damage for the rest of your life. It's a very contagious virus too. It passes from other animals to humans. So that's how it spreads. And it has to be studied in very specific conditions. They called level 4 security laboratory, something like that, which seems scary. Okay.
It's a bat virus, by the way, mainly.
It's what?
A bat virus.
Okay.
It's spread by fruit bats, if I remember correctly.
I saw news. I was searching like news about Nipah virus, and it also like it decimates cattle, horses, everything that goes in the way. It's really, really deadly. Okay. Uh, so one of the possible reasons why Nipah virus is so deadly is that um, it negotiates with the one of the uh, innate immunity mechanisms that all our cells in our body have. So this innate immunity mechanism is the interferon response or the cytokine production as a response to a virus infection. So after the first layers of defense, for example, getting into a tissue, when the virus really penetrates the tissue and get into the cells, one of the main defenses is the production, and the cells detect the virus inside the cell, detect viral particles, single-stranded RNA, and they started producing the cytokines that they release into the environment, and other cells can sense. And when they sense for example, interferons, TNF, and other cytokines, they prepare themselves to defend against the virus. One way of defending against the virus is, okay, I sense a cytokine, and then I produce a antiviral peptide that can destroy the virus and inhibit its replication. So this is one of the important layers of defense in our immune system. It's not the adaptive immune system, so it's not specific for a virus, and it doesn't remember which virus you got infected 2 months ago. So it's one of the quickest responses in our body, and it is very important to alert the immune cells. Macrophages, neutrophils, dendritic cells, they get activated and recruited once your tissue is releasing all those cytokines. This is called inflammation. So Nipah virus can negotiate with this system, saying, for example, oh, create a little bit of interference so I can penetrate the tissue better. Oh, and now shut down the interferon because now I want to survive. Now I don't want to alert your immune cells, let's say. Kind of a cartoonish way of describing it, but biologically, what's happening is that Nipah virus has 3 proteins, V, P, and W, that can either promote the production of interferons in the early stages of the infection and inhibit the spike of the interferons in the tissue in a short, medium term. So this diagram didactically explains what's going on. So first you have the Nipah virus entering the cell. It is detected by the cell. So hopefully you can see my mouse here. It indirectly activates the production of interferon alpha, IFN, interferon beta, TNF, which is tumor necrosis factor alpha, CCL4, CCL5, these are all cytokines. They are released, secreted into the environment, and sensed either by the cell and the neighbor cells. So looking at a single cell, of course, since this is a ODE model, it's not a spatial model, everything the cell produces, like the outer environment, which they call media, is the overall effect of all surrounding cells being infected at the same time. So it's not a spatial model. It doesn't carry details of this cell is infected, the neighbor is not. Okay, there is no such detail. So think of a single cell. The single cell releases the interferon and sends it back. When it senses, it activates a positive feedback loop system called JAK-STAT. So JAK is the sensor, and STAT, this feedback loop here, you can see that the arrows close in a circle here. This is the cell mechanism to amplify the interferon signaling. So it senses the interferon and induces the production of more interferons, and so on and so forth. It's more complex than that. Of course, you have there is in later stages you also produce SOX that inhibits the activation of the STAT cycle, and it shuts down inflammation in long term. So there are all sorts of interaction here. It's a very complex model. So enough of background. Here in talking about my notebook, I have a few guidance, a little guidance here on how to read this diagram because it's scary. Oh yeah, the last thing I almost forgot to mention is P, V, and W, they act mainly by inhibiting the STAT signaling pathway. So they inhibit the production of STAT, and they shut down the positive feedback loop that the interferon production needs to have that spike of interferon that causes the inflammation. So in the case of the Nipah virus infection, the true inflammation happens after you are almost sentenced to death already. All right. In the paper, now talking about experiments and data, real data, which I think is important. And of course, James would request that if I didn't put it here. What they did in the experiments was they took another virus, NDV, which is a Newcastle disease virus. I don't know what it is. I didn't search. I didn't lose my time searching for it. So it's basically a dummy virus, let's say, that they can genetically modify to include each one of the Nipah virus proteins that affects the, the internal cell innate immunity machinery. So there are three proteins, P, V, and W. So they take the NDV and they genetically edit the NDV to produce the P protein, or produce the V protein, or the W protein, each one of them separately. This is fabulous, right? This is such a wonderful work. I mean, you take another virus and genetically edit it to produce a protein from another virus, so you can study the effects of that protein in isolation. This is super clever. I didn't even know it was a thing. So yeah, congratulations to these guys here. And what you can see in the graphs.
I may point out that this is when there have been conspiracy theories about the origin of SARS-CoV-2. This has been one of the kinds of experiments that was considered as the region that you had the problem, because this is what's called the gain-of-function experiment. I'm not saying they should or shouldn't do it. I'm not going to talk about moral issues in this class. But you're taking a normally non-lethal virus, and now you are adding things that make it much, much more effective at infecting and killing things. And so if your NDV virus plus PV and W were to accidentally leak out of your lab, you might have turned a common and non-lethal virus into one that can spread and also kill large numbers of animals or people. And so these experiments are very informative, but you want to be exceedingly careful that those engineered viruses that you produce don't get out. And probably the reason they did not do NDV plus P plus V plus W was because then they might have engineered a virus that could spread easily, like NDV, unlike Nipah virus, and was as lethal as Nipah virus. So you could actually use this approach to take, say, a disease that spreads very, very easily, like influenza, and add genes that make it exceedingly lethal. So it's a quite interesting approach, experimentally. And again, I'm sure they were extremely careful, and that they were also careful about the ethical issues. But I just want to point out that this kind of what's called gain-of-function mutation experiment, it can be controversial.
And also, if you can produce a vaccine knowing the virus without knowing the infection, only knowing the virus, you can make the virus and the vaccine at the same time, and suddenly you have a weapon of mass destruction that doesn't destroy your own country because you vaccinated your population. So there are all sorts of mad applications, I can imagine. Okay, so let's go back to the experimental results. NDV, the plain NDV without VP and W proteins, are the red curves. And you can see that, so this is in dendritic cells, human dendritic cells culture, okay, in a culture. You expose them to NDV, pure NDV, natural NDV, and the dendritic cells respond with a spike in interferon beta, interferon alpha, and TNF alpha, normally, okay? Then you add P, what happens? The orange curve. Suddenly the response, the cytokine production is much lower. And then you add V, it's much lower. And you add W, is even lower. So they measure that. So the mRNAs they did I think it's called inverse transcriptase, something like that, to measure the RNA expression, the mRNA expression in the cells. And they measured the interferons using ELISA, which it's like a luminescence stuff. I don't know exactly. So the idea is, well, let's build a model that replicates these results here, that can match or fit the experimental results. And I kind of gave up explaining all of the equations because they are 14 equations and they have all sorts of interactions. So I just pasted the supplemental materials. But for you here, for the purpose of presentation, I will explain what's going on. There are some assumptions. So the most basic assumptions here are binding of interferon in the interferon receptor is very fast, which means that the time derivative of the tyrosine kinase 2 JAK1 is negligible. So I'll go back to the diagram so you can see what's going on. Interferon here, IFN alpha, IFN beta, they bind to the interferon receptor and they cause the phosphorylation of JAK1. So they are saying this process is very, very fast compared to, for example, the production of interferons, which is low. So you are comparing, in a sense, you are comparing a protein binding to a receptor in the membrane with the production of a new whole protein. So it's a very decent consideration. So they can take off the equation, the ODE for JAK entirely, saying that the time derivative is 0, so you just algebraically manipulate the equation. You do not treat as an ODE in your model. So this is called quasi-equilibrium, where you assume that a change in a certain species is so fast, the time derivative is always 0. So it's always at equilibrium, let's say. Quasi-equilibrium was assumed also for hijacking of JAK by SOX, stat phosphorylation, stat P dimerization, SOX production, IRF production, IRF7 phosphorylation. It's all sorts of detail that I really don't think it's worth going through all of them in the presentation, so I will avoid giving you too much detail. The next main assumption is that the presence of the virus inside the cell is modeled by letting P, V, and W increase linearly in time. So they do not model viral life cycle. They do not model viral replication, viral unpacking, etc. They only model the presence of P, V, and W, which makes sense because they're not actually using the Nipah virus. They're using another dummy virus that effectively just injects P, V, and W in the cells. So they let P, V, and W increase linearly in time as a way of saying, oh, your cell is now producing P, V, and W constantly because there is viral mRNA that encodes P, V, and W, and that's it. And input parameters, the initial condition of all species is 0 except for stat. And stat starts at 0.1 micromoles per milliliter. These are all the equations. So here you have the mRNA for interferon. It has a basal rate of production. It's stimulated by IRF, which is interferon regulatory factor 7, and it decays. So you can see this also happens for mRNA of IRF7, mRNA of IFN alpha here, TNF, and mRNA of TNF alpha. With the difference that TNF alpha, instead of having a linear promotion like IFN beta, is linearly promoted by IRF7, here you have Michaelis-Menten. This is also true for the CCL4 mRNA, which is the mRNA that encodes CCL4 cytokine, and STAT-M2. The other stuff is like mRNA of interferon beta will produce interferon beta. So eventually here, interferon beta, you see this Michaelis-Menten, it's just producing interferon beta, which is automatically being released in the environment and secreted. So they don't care about the different phases of production, secretion, etc. So they are modeling both together. This huge equation, 31 here, this is the consequence of lots of assumptions they made, and they algebraically manipulate the equations to come up with this as a result. So I will not explain what's going on here. Other species are promoted by something and decay, promoted by something, another thing, and decay. I mean, I don't think it's worth it going through all of them.
No, I don't think, especially today, we need to get to Elmer, so I wouldn't,
Oh,
I
yes.
wouldn't worry too much.
So the last interesting thing here to talk about is they have this IC, which is inhibitory coefficient, which depends on the proteins P, V, and W. As you can see, IC has multiplicative interaction between the proteins. P acts as a promoter, V acts as a promoter, and W acts as an inhibitor. And down here in the stat, they all act as inhibitors. So they basically shut down stat pathway, which is the positive feedback cycles that really creates the spike in interferon. And down here, you can see clearly that they model P, V, and W as being constantly produced. So they increase linearly in time. So I tried, so the main parameters here that they fitted to data are Vmax2, which controls interferon production and secretion, K12. So I kept this style of giving the parameter, explaining what it does. Since this is not installed in nanoHUB, you have to install the packages, so I have to do that now. I forgot to do it before. And this is the initialization of the program. So here is where all the functions, code, everything is initialized. And down here, you can see the buttons you can select to specify your simulation, the graphs you want to see, and the outcome down here. So yeah, it's done. Initialized, I have to run this the first time. And then I select, for example, IFN alpha environment. Norm stands for normalized. I guessed how they normalized, so this is not perfect. I guessed what they did. I'm not sure. And here are the default parameters. And you choose what variant of virus you want to test. NIV is all the 3 proteins being produced. and the VV is only V, only P, only W, and neither is just the normal NDV virus. So I select something and run the plot, and it gives the result. So as you can see, the IFN alpha environment normalized. So this is like 10 to the minus 5 what it could have produced. So it could have produced 1. So if I select here NDV, which is none of the proteins, and plot this again, it goes to 1. So this is my normalization method. Now I can, for example, select, let's see what happens with only V and plot IFN alpha. It decreases to 0.007. So this is 0.7% what it could have produced. I can do the same for P. P is a little weaker, 3%, and W is the strongest, 0.0002. And I did the same, so let me select NIV, all of them. The same for interferon beta, TNF alpha, IFN, mRNAs. Yeah, this is really, really low, but if you go to neither, I think yeah, I'm not sure if everything is right, but I can check specifically the interferon alpha case. But yeah, you can see with neither of them, mRNA for interferon beta goes to 1. If I check NIV, which is Nipah virus, all of them, you can see it goes to 10 to the minus 14. So it's basically 0 mRNA that encodes interferon alpha. Now these graphs here look very similar to the ones in the paper. I have to add the other results here. And basically, these species are the ones they showed in the paper. So these are the ones I am coding here, showing in the widgets. So you can select the parameters. My sliders are logarithmically scaled because this makes more sense in the case of what is going on here. So you don't want to compare 10 molecules being produced with 11 molecules being produced. You want to compare 10 with 100. So I chose to set a logarithmic scaled sliders, and you can vary all willingly. And the only problem here, let me close this, is that the person has to refer back to what each parameter does, reading this cell over here. And this is all. One thing I will probably try to do to make this more interesting is being able to check multiple of them, for example, 1, 2, and 3, and display all of them in the same graph.
I
This
think
is
having comparisons would be interesting, yes.
yeah, this is but I couldn't find a widget that allows me to select multiple. I will do that as a homework.
Well, or you could always display 2 of them or something like that. Right, you could always display the one with all of them turned on, the one with none of them turned on, and then say, now display the one with only one of the 3 turned on and compare. But that's a good an interesting question. I think here, and again, for everyone, it's tricky, especially when you don't have it all, it takes time to get everything running, which is, it's difficult but necessary to try to think of some exercises. In other words, change the parameters and see what happens. As you know from the homeworks, doesn't give you a lot to work with. Whereas if you say, try this particular value and this particular value and see whether something goes up or down, it's helpful to at least have a few initial guided exercises. And then once people have done that, maybe it's easier to explore. Uh, but it's a fascinating paper. Other comments for Pedro? I know we're already at 6:15, so I want to make sure that Elmer gets to talk. Elmer, go ahead.
But this is quite impressive program work. Could you show your code? Have you implemented all the 40 equations? And
Yep. Let me
Cool.
show you.
I think this will be quite
These are the 14 and the rates. So I implemented, like, for example, J1 is IFN is produced and the rate I specify down here where I organized the rate and the values of the parameters in a single box. And I do this for every single one of the equations. Yeah, this was a lot of work. I don't want to talk about it.
well. It's impressive, definitely. Yeah.
Two questions, Pedro, quickly. Can you, in the equations, the PV and W, I think they are, right? They modify both the stat pathway and they modify the IC. Can you remind me what the IC is?
Yeah, so I see in the diagram, I see is happening over here. Okay.
Okay.
IC is either inhibiting or activating the production of the cytokines directly, the production, while the other case, so let me go back again. So this IC is acting over the production of cytokines. So for example, CCl5M, you have IC over here, multiplying the basal production. CCl4M, you have IC here, multiplying both the basal rate and the promotion of TNF alpha. IC over here, suppressing production of TNF, and so on. In the case of STAT, so you have PV and W, they all inhibit STAT. So that equation down below is referring to this action right here, PV and W inhibiting STAT.
Okay, but for IC, P and V promote IC, and W inhibits IC.
IC is not a species, is a
Yeah.
coefficient.
Yeah.
And yeah, so yeah, the way you have to think about it is P and V, they promote the production of cytokines
Mhm.
here, and W inhibits. Okay, so what's going on is P and V promote the production of these cytokines early on, but they inhibit the positive feedback loop, which is the positive feedback loop is the single mechanism that spikes the inflammation. You need a positive feedback loop of sensing, producing, sensing, producing, sensing, producing, and doing that repeatedly to really have a spike of inflammation. So this promotion down here, it's like, okay, you produce a little bit more cytokines early on, but this is not enough to activate your immune system.
Okay. Okay, I think that makes sense. Okay, my second question is just, did you, I don't know, because you in your graphs you have P by itself, V by itself, and W by itself. Are there combinations of the two that you can try, like P and V and P and W?
It is certainly possible in the software end, but they haven't done it in the lab. So even though I thought, oh, I could do it, they didn't do in the experiment, so I kind of neglected this possibility. But yes, you can do this in the code.
Well, but that could be interesting. I mean, that's something you could easily add to your display if you're doing comparisons. You could
Yeah.
turn them on one at a time and see how they sum to each other.
Yes, yes, it's certainly possible. Yeah, it's a very easy thing. So let me just.
Oh.
Yeah, I mean, eventually in one of those, I think it's here. Yeah, so there are flags for each one of the proteins. So I can activate V and deactivate P, activate both, activate all of them. I can do all sorts of combinations.
Right.
So the values, oh, sorry, last question. They recreated this in the lab, all these like modeled scenarios. And did in the end of the paper, I guess, did they say it's like accurate?
They say that they don't say it's accurate. They say they agree, like one do what the other does, but I am very skeptical about the parameters, for example, because for example they plot the values as normalized by the maximum response. So you cannot directly compare the result provided from the model with the result provided from the experiment. There are all sorts of limitations, and the model itself is huge, huge. And think with me, how do you model this curve here, interferon beta, this curve here? It's like just an ascending curve. Do you really need all this to model 3 ascending curves? This one, and this one? So it is, in my opinion, it's an overfit. You have a huge parameter, 49 parameters to fit a few ascending curves. So
we're trying to model known.
That's true.
Right, that's the ideal thing. I think that's what biology Lotka search is.
So I wouldn't say it's like accurate. I wouldn't risk to say it's accurate, but it does job, and certainly it's a big challenge to work with this model. And it's good that they are working with a complex model because someone needs to work with a complex model. We have evidence, like enough evidence for every single one of these agents, let's say. So eventually, if you're ambitious enough, your model has to include all of them eventually.
I mean, I assume that this pathway without the PV and W was already studied and modeled before, and they're just trying to fit PV and W into the existing model, seeing where they can add inhibitions or promotions that will give them the curve that they got experimentally.
But even provided that, yes, it refers to a previously studied model that fitted some parameters to other data. Most of their parameters, remember, 49 parameters, only 5 of them are being fitted here in this study. So 49 parameters, the vast majority are guessed. They say, oh, this is a reasonable parameter from like physiological context. So you guess a parameter. Oh, this is like 10 to the minus 3. Oh, this is 1. This is 5. It's like many of the parameters are like this, you know. Just the exponent, and that's it.
Again, I'm not trying to be, again, I want people to have the time they need, but, and I think that the level of engagement and discussion is fantastic. But but I do want to make sure everybody has a chance to present.
Analysis of the Boolean generator network was what I looked into it, and it was a paper about the myeloid progenitor cell differentiation. And I was interested to re-implement this model in Tellurium, and I will come to that. So first, a little bit background, a refresher. I have already showed this slide in the introduction. So we have blood stem cells that are generated in the bone marrow, and that's the hematopoietic stem cells, and they can differentiate. Either they differentiate further in the bone marrow, and that's the myeloid system, or they go in the lymphoid system where we have the lymph nodes and so. And in the lymphoid system, there will be T cells, for example, and dendritic cells that Peter talked about will be generated. And this is part of our targeted immune system. And in bone marrow, we have the more unspecific part of the immune system, which are macrophages. And we have as well the red blood cells and the platelets that heal when we, for example, cut us, heal our skin. So focus on this myeloid progenitor differentiation is really on this myeloid side. I hope you can see the mouse pointer. And I have here orange highlighted the checkpoints that they as well had in the models, and that is described next. So the other part of the background are the Boolean generator networks. And then I talked about this as well in the last presentation, and we had more from that in our class. So I think I don't have to go into the details, but more or less the connection from here we have 3 genes, and the connection, how the genes are connected together is regulated over Boolean gates. And from that you can build transition tables, and from the transition tables you can derive the kinetograms and figure out in which attractor they will fall, or how many different subnets you get. So, and I was interested to have a bigger model that we can analyze in just a toy model. So the aims of my work was really, the first aim was taking the model that was published in the paper, translate it into Tellurium software. The second aim was to investigate the elements of the network and the functional motifs. So, and that's at the moment where I'm stuck. I was not coming totally to the end what I want to do. And then is as well, they have used in their paper, there are different timings in some of the, actually it was in one of the papers that was not exactly this differentiation paper, the paper before, but they can use with the software that they have different timings. And I would like to understand that, and especially this software that exists in SGVerse, that's in the ScanPy package, and I want to use this for my own work. And I really want to deep understand how they do that. Otherwise, I cannot really use this software tool. Good. So, short explanation about the generator network that was used. So, this is before we had this differentiation part, and this was the network. And actually, the interesting thing is this network is not totally saying the same as the function that they describe to every gate. There are differences here in this, how the June protein is connected. And they talk about that, and I don't know why they have not adjusted the model, but I think for my notebook, I really have as well to regenerate this graph and or show how it's differently. But that's what I myself oriented on, and it's as well what the software is oriented on. So it's this table that describes each of the genes that we have, and it describes the Boolean function and it gives as well some references about why they took these Boolean functions, and they think that the Boolean function have to be like this. So thinking about the class that we have, let's skip over the first two functions because they're complicated, but we see the fog1 to gata1 function. This is just fog1 has the same value like gata1, and then the ELK, the FLI, and the STLI function. These are exactly the gates that we had when we had the protein A and B, and they were autocorrelating and they were blocking each other. So in the Boolean logic, this is called an imply gate. This is non-implication gate, and this is A and not B. That is what it says, and it's exactly what these three equations describe. And this is one of the most common gates in biology, and is often used in synthetic biology. And it's totally not common in digital electronics, where we mainly have NAND and NOR gates. But on the end, it took me a while to figure things out, but it's all the same. If you want are down on the logical level, yeah. So then the gene, same, is a non-imply gate. The ERGNAP is as well an imply gate, but you have here an AND connection. So you have PUI1 and June that is AND connected, but then it's in the big picture as well an imply gate. The same for the PUI gene, but there you have CEPB alpha or PUI and then not GATA1 or GATA2, but on the big picture, it's an imply gate again. And you have SERB is different. So the GATA genes are different. The SERB gate is different. And even the last, the GFI is as well an imply gate. So they have all the same structure, and we will come to that when we go through the notebook. Oops, sorry. So, yes, so let's go to the notebook. Can you see that? Yeah. Okay. And I have as well to organize this a little bit nicer with the scrollers like Peter have, but more or less have the model loaded here. And this is exactly each of these equations that we had. And I could more or less just take the equation here that how we had them in class and translate them and put them there. And I did this for each of these around 20 equations. Then I set the basic parameter, and when I run the model, this is how the time course looks like for each of the genes. Now, this is now after I iterated a lot of the models, but of course I made as well some mistake when I really entered all of these equations. And what I did is I took each of the single genes, and I was really each single gate I was plotting. So this is the simplest one. And I have as well to say I translated, there is something called Hugo gene names, this is the consortium that actually gives the gene names. And there is something called Ensemble, that is where you can get information about the genes. And that's databases that I look into, and I think we should really stick to common vocabulary and use the gene names that are from the consortium suggested. Now, the problem in biology is that some of the gene names that are used are in some community much older than when we sequence the whole genome. So you have, for example, genes like ERBB2, which are commonly known, for example, in breast cancer as HER2. So all the biologists will this call HER2, but the correct name would be ERBB2. Now, the problem is when you bioinformatician and you have to retrieve data, you will find this gene only under the name ERBB2. So I really went as well through this whole model, and I changed the name to the explicit name because I want to later on link this to data, and there I need the correct Hugo name. And that's why some of these gene names will now be a little bit differently. So, but FOG1, what it's called, is ZFPM1, and it's similar to GATA1, and this is what I see. So I just go over GATA1 and have different values, and more or less I get always the value equilibrium that I set with GATA1, I get as well by ZFM1, so this was correct. And then we come to these non-imply functions, and I plotted them. So this is the time course, and this is kind of the gate that you can see. So you have, if GATA1 is high and FLI1 is low, then you have a high expression. This is exactly when it would be and, then it would be if GATA1 is high and FLI is high, then you would here have high, but it's exactly but GATA1 and not FLI1. This is how the regulation works. So then I just scroll over all of the genes, but they look, of course, all the same, because as I said, in biology, this is the basic gate. This is the gate that is around, and it's the same gate that we had in class. We called it there actually and-like gate, but yeah. Okay. You see, that's really the major ones. Now we come to an interesting one. So this GATA1. And you see that GATA1 is self-regulated by GATA1 itself, by GATA2, and by FLI. So these are OR connection. We sum them up. And then we have as well an AND connection with SPI. And we have the decay. So this is the Boolean way of, or the programming way of writing it. That's, you see there. Yeah. So, and interestingly, this is a time plot, and it looks to me like a bifurcation, and I haven't had time to really go into the or I haven't time. Took me too long to get really clear on this, to really go into the bifurcation diagrams and do the phase plots. But I want to do this because you can already see in the gate that here is just a whole gap and we don't get data. So it looks clearly like a bifurcation, that we only reach the higher level and the lower level, and when we have SP1 low, so that here there have to be a bifurcation, and then we have the data up there. And then another interesting one was this, actually as well, just a normal NumPy but itself. So SPRI1 is as well itself, it's auto-regulated. And that's why we get as well kind of a bifurcation here. So similar picture here. And then we come into, it's an NumPy plot, but it have here these AND connections. And here we get really an interesting time series. And this is the plot that I get up. It's as well self-regulated. So SEBA, I think it's self-regulated, isn't it? Maybe, yeah, it's a SEBA to SEBA and mapping. So we have and SEBA. So it's always when it's auto-correlated, or it's auto-mapping to itself, then I think then we get these bifurcations. So definitely have to do a phase diagram on that, and the bifurcation plots. And then GATA2 is as well more complicated, and we get as well quite interesting plots out. Okay, and that's actually really where I'm at the moment in the implementation. But yeah, I mean, there were in the end 4 papers that I really was in deep studying, and I want to show here as well what I still want to do the next week. Let's go back to the presentation. So I have the Ani model, I have the Boolean gates, I still have to go into deeper analysis from the single gates, the bifurcation, as I said, in the phase plot, and then I really want to go in this network analysis. And in their paper, so this goes actually into the Muir-Reed differentiation. So there are 4 major attractors, and 2 of them split really early away, and then they divide a second time, and this is more or less the upper part and the lower part. And according to the gene expression that they detected, they correlate this attractor to the different myeloid cells that are reached, and I want to be able to see the same in the plot. And this is one part of the study, and then what they as well had, and what I'm very interested in. So this is from the Wittmann paper, who first describes how you can actually really come from this Boolean model and compare it to real world data from the lab. And they used actually the K value that we always set to 0.5 to have different timescales. And in this model that they studied there, in this Wittmann paper, that's not the model that the Muir model that I'm looking at. They had these different, the dark box is a very slow scale, I think. Okay, and then a medium scale is T2, or was it one is slow, one is fast, and the medium scale is a general one. So they have really used these 3 different time scales, and they reach these time scales by setting the K value. Now, in the software from SIverse in SCMPy, that's how they describe the whole model in this text file. And you can see here the Boolean model. Oops, sorry. The Boolean model is described here. They set some parameter, they use Hill equations like we did. But then they have as well something called a coupling list. And that's where they each gene say how the gene is somehow coupled to each other. And in the software, then even if they reference this paper, nowhere they specify initial K values. And they have as well another constant that they describe in the paper, which is the decay is not always first order. They use a pi factor that they can as well adjust. And as well, this pi value, I cannot see where they do the default setting for this pi value. And I have no idea what this coupling value really is. So I reached out to them. They are on Tulip, which is similar to Slack, and I ask about that. I read this paper and I cannot see that. And so far I had no answers. This is from yesterday actually that I really wrote them, because I had first really to see if this not is in the paper, and I hope they answer me. And one part of the analysis would as well be to understand this coupling factor. I really would like to get this in to fully understand the software. These are the references to all the papers that I'm in deep studied. So the Krumsky paper was the first one that was published on the model, but mainly the Krumsky 2011 paper is the one that this Muir-Reed differentiation is discussed. Then we have the Wittmann paper, who really was the one who takes about the formality to transform the Boolean model into this continuous model in an automatic manner. And then the Wolf paper is just a scampy implementation that is the software that I would like to use for my own research work. But yeah, so that's where I'm at the moment in my project, and I know that there is still things to do. Thank you.
Comments or questions on this? Anybody? I mean, I think you're seeing something that you expect, which is anytime you have auto activation, you expect that there'll be bistability. And the only thing you're seeing is that depending on how you modulate that feedback, you either get bistability or you don't. So that's a very simple bifurcation. You could then explore what happens when you change the rates of the well, in the case of a bistability, you really only have one feedback loop, so there really aren't any rates to think about. But I think one thing you might want to try to think about is how you could make this into something that somebody else could understand. At the moment, you're working to learn how to do this for your own project, which is great. But I don't know how helpful this would be to somebody coming to it from outside. And so one thing you could think about is, if you go back, why don't you show your diagram again, the big circuit diagram. So you could start out with just say the bottom two negative, the cross-coupled negative feedback loop. You have ERG-NAB inhibits GIF1, GIF1 inhibits ERG-NAB. And you could just look at that two-component circuit and show what happens. In that case, when you change the K values, and K doesn't change the rate, it changes the equilibrium. It's the Vmax that changes the rate. Now, if you don't go to saturation, then the K can also be used to change rates, but it's not really the right way to do it. But you could take just that cross inhibitor, which is a toggle, and study how that works as a Boolean network and then as a Hill function. And in that case, as you change the Basel rates and the Ks, you'll get some interesting effects. And then you could take a circuit with 3 components. Let's think about the simplest one of 3 components. There's a couple. There's a GATA1, GATA2, and then whatever is hiding under your knockout there, where you have 3 latches that cross-inhibit each other. And you could study that circuit by itself. Or you could study the GATA1 EKLF FR1 feedback loop, where you have a latch at the bottom. This is the bottom left. Look at the bottom left. All the way there. You have FLE1 inhibiting EKLF, and then so you have a toggle there. But FLE1 is cross-promoting GATA1, and GATA1 in turn is promoting ELF. That one will have interesting, that triple will have interesting, and you have GATA1 promoting itself. That one will have some interesting differences when you go from the Boolean network model to the reaction kinetic model. And so I think that if you start with the very simple ones here, and then build up, you can actually understand in some detail, in a very didactic way, what's going on. If we'd had more time in the class, that cross-inhibitory toggle is the next thing. In fact, if we had had time today, I was going to do that toggle circuit. That was actually the next thing I had on my agenda, was to do it to show you how that toggle circuit works, where you have the cross-inhibition. And you did it in homework, as an example.
Yeah.
And so I think that and this came up, I think, also in Pedro's talk to some extent, where Pedro had there were a couple of core feedback loops. There was that TNF alpha feedback loop, auto-excitatory feedback loop, and then there was also that JAK-STAT loop on the right-hand side. And so if you want to understand how these circuits work, it's helpful to try to break them up into modules. They're not really modular because the coupling is very strong, but you can get an intuition about what's happening. Anytime you see something like GATA1 auto-activating, that's going to be bistable. Anytime you see GATA2 auto-activating, that'll be bistable. If you had nothing else, if GATA2 is low, it stays low. If it's high, it stays high. Anytime you see cross-inhibition, like the two at the bottom, FLE1, EKLF, you're going to have one or the other one turned on. You can't have both turned on at the same time, at least if the coupling is strong. If you make the coupling weaker, then you can potentially have them coexist. And so one can then start with those basic two-component modules and ask the question, if you have a three-component loop, you can have two possibilities when you have a three-component loop. You could either have compatible circuit. Suppose that you have A inhibits B, and B inhibits C, and C inhibits A. Well, you saw that with the Boolean circuit. In those situations, it's frustrated. You can't have all of them satisfied, because two of the neighbors have to have the same value, right? You can't, if you have three A, B, C, and you want A to be different from B, you want B to be different from C and C from A, you can't do it. You can't do all three at once. That's called frustration. And so what will happen in a Boolean circuit is that will oscillate. Each time you run, at each time step, it will go to a different state. One will turn on, and then two will turn on, and then three will turn on. If you go into a reaction kinetic model, depending on the parameters and the time constants you pick, either you'll get some intermediate state where they all go to some middle value, or you'll get something where they oscillate. And so you can actually understand these motifs, at least the simpler motifs, like the two-component and three-component motifs. You can do a pretty complete analysis of what's happening with those. Now, when you put a lot of these circuits together, then it becomes more complicated to understand how they interact. But I think for the purpose of producing an example that would be of interest to people looking at that at the notebook, and also for learning for yourself, building up from these simple circuits and understanding how they work one at a time is not a bad place to start. And so I think you did a nice example at the beginning where you talked about one or two very, very simple ones. But I think one could go a little bit further with this and ask that kind of question. It's a little bit, you could do the same thing, I think, a little bit in Pedro's example, where he also started with quite a complex example. In Pedro's example, you could ask the question, if you knocked out one element of that circuit, what would happen? Would it prevent the inhibition of the interferon pathway if you knocked out that element? Or if you upregulated an element, could you overcome the suppressing effect of W, P, and so on? And if you can do that, then you can actually come up with drug targets. You can use a model like that to identify drug targets. And as Pedro said, you don't necessarily know from the beginning that the parameter values are right, but you do know that probably the direction in which things are being pushed is right, or at least likely to be right. And that's in itself interesting. You say, well, if we were able to knock out or reduce this component, then Nipah virus would be less effective at turning off interferon. Well, that gives you a drug target. Gee, if we turn this on more strongly, Nipah virus would be less effective at turning off interferon. That gives you a drug target. Now, you still always have to go back. These are always phenomenological models. You always have to go back and do it in vitro or in vivo to see whether it works. But at least you can have a rational understanding of what's likely to happen. And so in the model you have here, you can ask the question. Here you've got a question. If you knock this out, what happens to your differentiation state? You could go in and ask that question, knocking out probably not each one of these because that's too much to do, but you could pick a couple that you think are interesting, knock them out or knock them in, turn them on all the time, and then ask the question, how does it change the output? And compare that between the Hill function version and the Boolean version. So Another thing, of course, with all of these Boolean models is that we know that these things are stochastic, and so that they flicker. And so that's another thing one could do. I don't think for the purpose of this course, we never really got to look at stochastic models, at Gillespie simulations. But those are also quite easy to do in Antimony. It's just change simulate to Gillespie and you get the stochastic version. And that particular latch or toggle, which you see at the bottom, in the toggle in the deterministic case, you either have FLE1 turned on and EKLF off or vice versa, and that's it. Once one is on, it's on. That stays there forever. In a Gillespie model, it'll flip between the two states, one on, one off. And the rate of flipping depends on the coupling strength and the noise. And so they're actually sorry.
Because this is what the software takes. So so this is what the software takes into account.
I think you may be muted.
Okay, so this is input for the software, the compiled software, and they have this coupling list. And I wonder if this coupling that you said about with Gillespie, if they use this, because I still would like to understand what this coupling here is. But I was not just because you talked about coupling and the Gillespie algorithms, and yeah. Nice mute again.
Since I don't know this software package, I mean, one reason I like to stick with the packages I know is because I know what they do. And so I generally wouldn't use a package where it has coupling strength. Clearly, this is something to do with these are representing the arrows in the diagram, and they're representing the probably. My guess is I don't know. Again, if I had had a chance to do the toggle, I could have shown you how you adjust coupling to it.
Maybe we'll figure out, but what another thing I think would be interesting and possible to do is what I said about. I would like to graphically display transition tables, because I can really make a transition table of all the genes, and I can show how they change when I run the model. And in the Boolean model, or as well a version that is the relaxed model, the more relaxed model, and as well to find these state cycles. I mean, now I have the whole model, and you could really figure out the state cycles, and that would as well be making. It would give, I mean, we have done this basic model, these basic elements, and we have worked from a basic element to a more complex element. What I would be interested is how they play now together, right? How you really get to the whole process that's going to an attractor. And I mean, we have done the phase plots where we saw that sometimes a big way is taken till you end up in your attractor. But I think we don't have done this one that you see in CNT.
Well, we've done oscillators. We didn't only did one, but we did an oscillator example in class. Wasn't the nicest oscillator.
It's not an oscillator as such. It's really a whole kinetograph. So it shows every state and how they are connected. I can maybe after the class show.
No, I understand what you're talking about, Elmer, and these are certainly possible. In other words, you can definitely do all of these things.
This one. Let me quickly show this one. This is from. So this was one of the homework that we. No, it's not one of the homework. but we were studying this. I'm sorry. We were studying the A by A is blocking B, B is blocking A.
Right, that's the toggle, yes.
Yes, that's a toggle. And more or less when you really translate this into a digital gate, that is digital gate. And it's called flip-flop or a latch, right?
Yes,
And it's an SR latch, so you have set and reset. And this is the transition table, and you could display this transition table with the different values that you have, either Boolean or as well the continuous values. And when you follow this one, and when you really go at the kinetogram, you will really understand why you end up in one of these memory states, or why you end up in an unstable state. Just when you look at the kinetogram, and this is when I really digged into the transition table and this kinetograms, then I understood why the toggles really works. I was still confused what we learned how we look at it in the class, because there was not really a set and reset that really drive it back in how it's done in electronics to understand how it really works in biology. And that is what I would like to do as well with the complex model that we had. That's a little bit more than just toggle switch. That would be what I would be interested. I don't know if this helps other people. That's true. I don't know if it's useful for other people.
well, I think, again, I think if you find, I mean, I had a whole several slide decks on Boolean modeling. I have to say I got rid of them because people who came to this class generally didn't find them very useful. In other words, it depends on your background. In other words, if you're used to logic gates, then trying to reduce the biological complexity to logic gates can make it easier to understand. If you haven't seen logic gates before, then you're simply taking one unfamiliar system and saying that it's analogous to another unfamiliar system. And so I'm not saying that the logic analysis can't be quite useful, but it depends a little bit. But whether it's useful to you as a learner depends on your background. And so if you come from a situation where you've done a lot of logic, then that's great. If you haven't done that before, then of course it's a little bit more challenging.
Yes.
Okay, I think you've got a lot here, but I would try to at least illustrate a couple of these simple ones. This example that you have on the screen is a good one. So that somebody who looks at your notebook can learn how to build up a more complex auto activation, where A activates B, B activates C, C activates A, is reasonable. One with the one inhibitor, A activates B, B activates C, C inhibits A, is another one that's interesting. That's also frustrated. And you could get a lot of very complex behaviors by playing with the timing. Even a feed-forward network where you just have A to B to C just by itself, where if you have A goes to B goes to C, A goes to C directly, that can generate pulses. It could generate all sorts of interesting effects. It could be a filter of various kinds. It could be a low detector, high detector, depending on the timings and the values of K and Vmax. And Herbert has a whole chapter on that, the feed forward, the incoherent feed forward network. So those are all there. I don't know what to say, except that maybe this should be 2 semesters instead of 1 semester to cover this material, because really it's taken us a semester to have the basics. And now it's frustrating. We're at the last lecture, and we have the opportunity we'd have the opportunity to start playing and building interesting things, and we're out of time. And so
I mean,
I
for
hope
grad
at
students,
least
it's
you
maybe a
have,
bit different because we can keep on using
Right.
what we learned. I
Right.
think Pedro will as well use what he learned.
right. Well, I hope that people have learned enough tools that if they're interested, they now, when they read a paper, they won't be intimidated by it. If they look at the mathematics, it won't be so unfamiliar. Well, I'm
But okay, I understand the critics. I have to do something as well that is useful to learn from other from notebook, and I will do that. Yeah, definitely.
well, I just, I mean, you don't have to do anything, but I would recommend that it would be probably helpful to try to do something like that.
Yeah.
So I guess we don't get to talk about two-dimensional bifurcations this year. Maybe I could show you but do you want me to give 10 minutes and tell you the results?
I would love to,
Fine.
but I don't know what with the other people is.
Okay, well, people can leave. So I'm not going to take very much time. And it's not really set up to be gone through. To higher dimensional stability. Again, the basic concept of stability is the same. You start with a fixed point, you move some distance away from the fixed point, you ask the question, if you evolve it in time, do you get closer or further from the fixed point? The thing that makes things a little more complicated is that in higher dimensions, the displacement isn't a scalar number, it's a vector. And the length of the components in different directions could evolve at different rates. And so your dynamical equations then have the form of dN by dt is equal to some rate equation, which is a function of the fixed point plus the displacement of the vector. And you have to do a Taylor expansion. And the Taylor expansion in arbitrary number of dimensions is a bit of a mess. But the linear term of the Taylor expansion in arbitrary dimensions is just the Jacobian. That's the first order derivative, partial derivative of the functions with respect to each variable.
It comes all the time. Could you
Yes.
say some words about this Jacobian because it's
So your first, suppose you have n equations and n variables. The Jacobian is the derivative of the first equation with respect to the first variable, the derivative of the first equation with respect to the second variable, the derivative of the first equation with respect to the third variable, so on. And then that's the first line. The second line is the derivative of the second equation with respect to the first variable, the derivative of the second equation with respect to the second, and so on. So it's just putting out all of the partial derivatives that are possible in a matrix. And so it's all of the first derivatives. And if I had more time, I would walk you through all of the linear algebra here. But the question then is, what is the length of what happens to your little displacement vector? And it winds up that the displacement vector is time times the exponential of time times the Jacobian matrix times the initial vector. And it may seem strange to exponentiate a matrix, but in fact, you can do that. Exponentiation can be defined in terms of its Taylor expansion. And if you have a square matrix, you can exponentiate a square matrix. And that looks like a mess. But in fact, if you ask what the length of the vector is, what the length of that vector is, the way that vector's length evolves in time is given by the eigenvectors of the Jacobian matrix, the eigenvalues and eigenvectors of the Jacobian matrix. And I don't have time now to walk you through the arguments about the how the vector, well, I guess we can do that. Suppose that I have an arbitrary vector. The eigenvectors are the vectors which under the Jacobian matrix don't change direction. So the Jacobian matrix applied to an eigenvector gives you the same vector, either longer or shorter. It doesn't change the angle. And so if I analyze my initial perturbation in terms of the eigenvector, I can then expand the in each eigenvector direction, the length changes proportional to the eigenvalue, and I can then calculate my new vector. And if you've done linear algebra, you've done this many times, but the result is fundamentally that you can now calculate exactly what happens when you apply your operator to your initial displacement for an arbitrary displacement. And what you find is that if the all of the eigenvalues of the Jacobian matrix are negative, then the fixed point is stable. And if any eigenvalue of the Jacobian matrix is positive, then the fixed point is unstable. And the divergence of the distance from the initial position increases as the e to the time times the value of the largest eigenvalue. And so that's the fundamental result, is that if you take the Jacobian matrix of the rate equations and you calculate their eigenvalues, if any eigenvalue is positive, the matrix is unstable. Now, the fixed point is unstable. It winds up, there's a lemma which says that that's equivalent to doing the trace of the Jacobian matrix. If I just sum up the values of the eigenvalues along the diagonal, excuse me, I said that wrong. If I just sum up the values of the diagonal of the Jacobian matrix, and that value is positive, then that tells me that there is at least one positive eigenvalue, and therefore the fixed point is unstable. And so I don't actually have to evaluate the eigenvalues. I just calculate the Jacobian matrix, sum along the diagonal, and that tells me whether my fixed point is stable or not. And so it's actually quite simple to determine whether a fixed point is stable or not, even in high dimensions. All right. Now, there are some additional complexities, which I'm going to go into a little bit here, which is if you're in 2 or more dimensions, you could have a positive eigenvalue along one eigenvector, in which case you're unstable in that direction, and a negative eigenvalue along a different eigenvector, in which case you're stable in that direction. The net effect is that if you're unstable in any direction, you're unstable, but you could be unstable in different ways. And this shows the example of that. I could have, in 2 dimensions, I could have 2 positive eigenvalues, in which case I'm unstable in both directions. I could have one negative eigenvalue, one positive, in which case I'm stable in one and unstable in the other. And I could have both stable, in which case I'm stable. One result of this is that you go to higher and higher dimensions, you have more and more possibility of instability, because if any eigenvalue is positive, you're unstable, and you have n eigenvalues. If you have n dimensions, you have n eigenvalues. And so it's an and. If you want stability, you need A and B and C and D all to be negative. If any of them are positive, it's unstable. If you want to know the actual values, you have to use the determinant. And I'm not going to do the determinant now. But if you remember determinants involve square roots. And that means that you can also have not only positive and negative eigenvalues, but imaginary eigenvalues, complex eigenvalues. And those are related to the determinant. Those are given by the determinant. If the eigenvalues are complex, then the stability is given by the real part of the eigenvalue. But if you have a non-zero imaginary part, then you have oscillations. And so if you have negative eigenvalue with a positive complex component, then you're going to get a decaying oscillation. And there's a variety of situations. It means that you can actually classify all of your fixed points based on two things, at least in 2 dimensions, on the value of the trace of the Jacobian matrix and of the determinant. And so, and I'll skip to the end here, you have a variety of different orbits that you can get. But you get a final picture, and this is the last thing I'll show for the class. And I'm sorry to be stepping through these. For each combination of positive and negative trace, real and imaginary determinant, you can get different shapes. And you can put these down on a picture like this. It's not the prettiest picture. If the trace is positive, you're unstable. If the trace is negative, you're stable. If the determinant is positive, you get spirals. If the determinant is negative, you get saddles. And so that gives you a classification of all of the fixed points that are possible in 2 dimensions by evaluating the Jacobian matrix and then finding its trace and its determinant. The one thing that we've left out here is that you can also have limit cycles. You can have periodic oscillations. And then in that case, you have to analyze, are those oscillations themselves stable or unstable? You're not coming back to a single point. Do you come back to the cycle or not? And you can analyze the stability then of the cycle as well in 2D. So that's something that you don't have in 1 dimension, the oscillation. And that particular bifurcation has a special name. It's called the Hopf bifurcation. And you could ask the question, is that all there is? And the answer is that in 3 dimensions, you have 1 additional bifurcation. But that's it. There isn't anything more. Even in 20 dimensions, you don't get any more than that. And so that would normally take 2 hours to go through. So I appreciate that you're not going to be able to pull all of it. But at least you've seen some pictures of it, and maybe some of the terms will be familiar if we come back to it. So, okay. So we've raced through a few things about stability in higher dimensions. I appreciate your patience with just seeing those slides flashed, but I really do think it was more important to have you have the time to present and discuss your work than for me to give a lecture. And if people are interested, the whole slide deck is there. So you have the ability to walk through it slowly. And actually, Herbert's chapter on this is quite good, if you like. So thank you very much. We will have people present revised versions of these projects next week. Please try to remember that we have to get through 4 of them. So we have to fit them all in. So I understand you have a lot to talk about, but try to be balanced in your presentations. And I want to thank you for your patience tonight.
Thank you.